Now I'm going to go over many of the actual techniques, the equipment that we use to analyze biomarkers and the isotopes that we're interested in, and how we use these to get an idea of paleoclimate, paleovegetation, things like that. There are three main techniques that we're really interested in for not only biomarkers, but also residues of stone tools, or even residues of pottery if you're doing work in more modern context. There's been a lot of recent research looking at pottery as well. But the first one that we use is gas chromatography. And gas chromatography is great for identifying compounds themselves, biomarker compounds, and their abundance, and their relative preservation in sediments from your sites. From there, that can only tell us so much, the source of biomarkers, in their preservation. But if we're interested in climate-related studies, we need to understand their isotopes. So we have some techniques that look at isotope mass spectrometry. And then finally, pyrolysis is the third technique, another residue technique that's being applied to paleoanthropology more and more now after it's been shown to really understand those materials that are 400 million years old. They did a lot of pyrolysis with those stuff. Okay, so gas chromatography, mass spec. Most of you are probably familiar with this technique and why it's important for analytical chemistry. Whether or not you're a chemist, it is really great for a lot of the sciences. And it's really broken down into 6 steps. And the first one, gas chromatography, really is separating compounds by molecular weight. So lower molecular compounds like methane, C4, is much lighter than something like C27, one of our N alkanes. And it'll separate out of our sample earlier depending on its size. Then we have to transfer these compounds to the mass spectrometer itself. And this helps identify the abundance, the compounds themselves. And we do that with ionization. And then we analyze the mass itself. And then we detect the ions from the electrons to get us a readout. And then we process the data with the computer. And a general schematic, and this could be kind of confusing in general. So our sample is usually injected. Typically, you take your sample and you turn it into a liquid, and that sample is injected into our GC. And the GC itself is equipped with an oven and a coil. In the oven, you temperature program to, 300, 315 degrees Celsius. And that's enough, that temperature is enough to break apart most compounds that we're interested in through time. So over time and through temperature, your compounds break out. It is then transferred through our analyzer where those ions are picked up and identified. And then the abundance is typically brought out on our computers, our readouts. So GC-MS is great for basic analysis of equipment. But in order to really understand hydrocarbons or N-alkane biomarkers, we need flame ionization detection. So it's another type of GC that's coupled with a mass spec that uses helium carrier gas to identify just hydrocarbons. So if we're interested in N-alkanes, which are just carbon-hydrogen bonded to each other, this technique is perfect for analyzing that because it just picks up hydrocarbons. It ignores other compounds, things with, or inorganic compounds, like CO2, for example. It doesn't register that in your output. So if we're interested in N-alkanes and biomarkers, the FID is the best approach for analyzing these compounds. And when you do analyze your compounds, what the output is this. So here are N-alkane compounds themselves, everything from C21 to C33. So we have 33 carbons for this example, 33 carbons and 64 hydrogens or whatever. And you get a relative abundance. So the size of the peak is showing you how abundant that compound is in your sample. for terrestrial plants, vegetation growing on the land, trees, shrubs, grasses, we expect to see a high range between C27, 29, and 31, and sometimes you even get a high peak for 33. Whereas the lower samples are coming from aquatic plants or emergent plants that live near shore. Once we have these compounds, so this is a general flame ionization detector output. We call these our chromatograms. Let me go back one. Well, anyway, so our abundance here, and this is through time. So as you see that the heavier elements, I'm sorry, this is blocking the way, but the heavier elements are separated out later because they are heavier than the earlier stuff. And that's really how we quantify what it is we're looking at, what our samples are showing. C27 and 29 are trees and shrubs, whereas C31 is typical of our grasses. And C31 is really great for identifying C3 versus C4 plants. It's a direct identifier for those, whereas the other compounds can be influenced based on those themselves. Okay, so once we have our compound abundance, it's a really simple setup for analyzing the N-alkanes. We then really need to know what isotopes are comprising those individual compounds. We read these a few different ways. So again, like I said earlier, this symbol here means delta, which is a ratio. And here's the formula here for the ratio between the heavy element and the lighter element. And when we have a more positive value, that sample is enriched with the heavier isotope. So if you have something closer to 0, you're typically enriched with carbon-13 or hydrogen-2. Typically our values are in negative range, so the more negative value, we're more enriched with the lighter isotope. And this is all relative to a standard. So the standards for hydrogen are Vienna standard mean ocean water, whereas carbon is PD belimonite. It was a paleogeologic outcrop in the US. And now we use these to identify. So why this is important? When we're looking at our compounds, our C27, 29, and 31, we're really interested in this output. What it means, what is the ratio between the heavier and lighter isotope. And like I mentioned before, if we're depending on our source of biosynthesis production, we can get an idea of where they're coming from. If it's an algae, if it's an aquatic plant, or a terrestrial plant. And if we're really interested in terrestrial environments, in what is happening in the ecosystem where our hominid relatives were living, we want to focus on these larger chains, these C27 through 35. Okay, so this is a little muddled, but how we really distinguish isotopes from, say, the compounds themselves. So if you're doing this technique and you first use your GCF ID and you identify your abundance, you then run another type of mass spec that analyzes just the isotopes. And in this case, I've just selected out a few, C27 through 31. And you can see our negative values here for carbon. So the more negative you get, the less of the heavy isotope you have. The more positive positive value you have more of the heavy isotope. Now, of course, when you look at just the numbers, it can be really confusing. It might not make a lot of sense. So we like to obviously plot them in data. When you plot them, you should notice that they're really synchronized. So all of the values, they have sort of this negative trend at the bottom. They're decreasing in their heavy isotope. And then they sort of rebound and go more positive before getting more negative again. What this is showing us is that our values are highly synchronized. This is important because if you're interested in a particular environment, you need highly synchronized data. Otherwise, you know your sources are totally different. If C27 here and C29 looked completely different, you know you had an issue with your original sample or the technique itself. If they're unsynchronized, it can cause a problem. It can show you that your data is maybe not what you need for actually giving these ideas on paleoclimate. what's familiar, so if you go back, oh, I guess we won't go back, one slide. C3 plants typically range from -20 to -37 per mil, whereas C4 plants go -10 to -19. So really with your FID, you can, with your isotope ratio, you can get a good idea of C3 versus C4 plants. But these do get muddied by the biosynthesis, whether or not your plant's a terrestrial, aquatic, or emergent plant. That's why NLK and C31 is really great for discriminating between C3 and C4. And this is a direct proxy. A lot of the times our proxies for C3 versus C4 come from ocean sediments or from teeth, you know, what we see, the isotope composition of teeth. And those, because of fractionation, might change through the organism, and it might not be as direct as what we actually need. And this final technique that's really emergent in paleoanthropology is pyrolysis. And what that really is you take your sample, you heat it really high, like 600 degrees Celsius, and the fragments, the molecular fragments that result are how you analyze and say what you're looking at. This is great for looking at the conservation and abundance of samples. So those examples that we looked at earlier from 400 million years ago, a lot of the work they did was pyrolysis. It's a really simple technique, very little sample preparation. And it gives you a ton of information with your samples. So here on our right side, we have our typical mass spectrometer. The GC is over here, the mass spec oven is here, sample injection. The pyrolysis is just a small external piece of equipment that just replaces the input over here, and that's all you need. I mean, yes, it's expensive, yes, it takes a lot of the initial investment, but once you have it, it's really easy to get going and running samples. Like I said a minute ago, what the benefits are, well, there's very minimal sample prep. You take your sample, less than a microgram, or a milligram, excuse me, you place it into the pyrolysis itself and you turn up the heat. You turn it to 600 degrees and then you get your resulting chromatograms. The chemical fragments are easily understandable once they are run through, and then it can give you both quantitative and qualitative data for what you're looking at. So some of you who don't do archaeology or paleoanthropology, this application really is applied to many different fields, from forensics all the way to artwork. So if you're interested in some of the original art pieces, you can look at paint from some early art to see whether or not it was updated at any point through history. Things from DNA, identifying animal versus plant DNA on things, on samples. And really, it got its start in petroleum. The petroleum industry is really what advanced pyrolysis studies. And at our university, we have a huge research group doing a lot of work in petroleum, and they really pioneered some of the techniques for pyrolysis. Again, it's a very simple technique. You would inject your sample, usually a solid sample. You could do liquids at times. It gets run through the oven at really high temperatures. So you inject it at a high temperature, about 600 degrees, and then through the oven it gets lesser temp, 300 or so. And we usually use a 60-meter column, and this helps as the samples are being run based on temperature. They get separated out through time, and then we get our detector, our compound abundance. Heavier items show up later in our peak, where the lighter elements come early on. So what we're interested in is residues on stone tools, and whether or not we can remove the residues and then analyze them for the chemical components. And this is really how simple it is. So if we can successfully remove residues from stone tools, the sample itself just gets placed right here. This is a 2-millimeter quartz tube. I mean, it is really a pain to get the sample in. You have your glass wool as just kind of a barrier, a protector, so the sample doesn't fall out. And then this tube here gets placed into the probe, and then that goes into your machine. And then you combust it to 600, and you get your resulting pyrograms. And over time, this is a typical chromatogram will look like this. So as the peaks come out, as you're going through time, you get your different compound abundances, the relative intensities. In this case, we have our alkanes here at the end, but then we're interested, in this example, we have some other more complex compounds, levoglucosan, benzene, phenols, polysaccharides. And then from there, you know, this will give you a general idea of what it is, your state of preservation. Do these compounds match up from 400 million years ago to modern examples? How well preserved are they through time? And then from there, you can go and look at more detailed analysis, like isotopes, for example.